So this is a video that gives an explanation of how to create your figure 1. So figure 1 is a component of your genes and disease final poster. So here what I'm showing on the screen is on the left hand side here I have the instructions. So these are detailed instructions about what should be included in your figure 1, and then on the right hand side is an example of what your figure 1 should look like when it's completed. So what I'll do is walk through each of these components so you can kind of cross-reference both the instructions and then what that looks like physically in the actual figure 1. So if we start on the right here, if we look at the very top, there should be a heading or a description for your figure 1. So this says figure 1, the GAA gene with RNA and polypeptide products. So you can basically replace this part here with whatever gene is appropriate for your particular gene and disease. And then the part that's shown here, those are the references. So in the heading here is where you want to reference any resources, NCBI, GHR that you used to create your figure 1. And remember, we're using a numbered reference format for your genes and disease project, so that's why these are numbers here. So you'll have at the end section of your poster a numbered list of references, and then these numbers correspond to each of those references. So that's the heading that you want. If we then move down, the next component here is the gene location. So if we look over here, the description for gene location says to include the chromosome number, the arm, and the position. And remember, this is the format that we talked about when you're giving chromosome positions. You have first the chromosome number. So if we're looking at here, 7q28, 7 would be the number of the chromosome. Q refers to which particular arm of the chromosome it's on. Remember, it's either P or Q. And then the number there indicates at what position, relative position on that particular chromosome. So in this example here on the right, we have an image here that is showing the chromosomal location for this particular GAA gene, so where that GAA gene is located. So for the gene location, you want to make sure you're giving the actual written out form of where that is, and also an image with this yellow arrow indicating where specifically your gene is located on that chromosome. And so as we move down here in figure 1, what we're doing is essentially going from broad to more specific information. So we'll start with the actual gene location and then we'll narrow that down each step of the way. So the next part we have here is the gene. And so if we look at what we are supposed to include for the gene section, we want to include the length of the gene and make sure you include units. The units here are base pairs. So if we look at the heading here, gene, the GAA gene has a length of 18,342 base pairs and has 3 transcripts. So you also want to explicitly list not only the length, but also how many transcripts it has, in this case 3. And then the image that's shown here is a diagram from NCBI with the introns and exons displayed. So remember, each of these filled in boxes here, those are the exons, and then this is the number of transcripts. So it says we have 3 transcripts. That's why we have 1, 2, 3 green lines shown here. And on the next slide, I'll give a bit more information about how to obtain this particular image here. So that's for our gene. And then if we move next, we have our primary RNA transcript. So the gene is the DNA level, right? So from the DNA, we then go through the process of transcription to get our primary mRNA. So that's what we have next. So now if we look at our RNA structure section instructions, we need to include the length of the primary RNA transcript. So you can show the RNA transcript basically as a filled in colored box. So it doesn't have to be fancy, but you can just create that as a shape in PowerPoint or something like that. So you're including that colored in box, and then you're including the length of that primary RNA transcript. Again, make sure you're including the units, in this case, which is base pairs. So we've created a primary RNA transcript, and then through RNA processing, the next step along this flow from DNA to RNA to protein is our finished mRNA. And what we want here for the mRNA, we again want to include the total length of that mRNA. Again, make sure you're including the units of base pairs. And we can show this mRNA the same way we showed the primary RNA transcript, as a colored filled in box. And then in addition to showing the full length of that mRNA, we also want to divide it up. My dog has just come into the room and is staring at me. We also want to divide that up into the 5' UTR, so we're giving specifically how long that particular section is, the 3' UTR, how long that section is, and then the coding region that's in the middle. So for mRNA, you're giving both the total length as well as the subsections that are shown there. So then once we go from our mRNA, we do translation at the ribosome. Then what we make is the polypeptide. So if we look at the polypeptide product there, we again are including the full length, but now note that the units are different. So since we're talking about the polypeptide now, our units are AA for amino acids. So we're again including the full length, and we can again illustrate that polypeptide as a shape, a filled in box. But we're including both the full length of the polypeptide and amino acids, as well as any potential subsections. And if you note here in the instructions, it says if applicable. So a signal peptide is something that a protein has if that particular protein needs to be exported from the cell, or it needs to function in the lysosome or something like that. So if your protein isn't supposed to go to one of those final destinations, it won't have a signal peptide. So not every one of your proteins will have that. Same with the pro-protein sequence. Some proteins have that, like insulin, for instance, has a pro-protein section because it wants to stay inactive before it gets to its final destination to do its job, but not all proteins have that. So if your particular protein doesn't have a signal peptide or a pro-protein, you don't have to put that on this diagram here. But if it does, make sure you're including not only the full polypeptide sequence, but also the signal peptide, the pro, and then the mature peptide sequence. So breaking that up into these 3 different chunks, just like we did for the mRNA. So that's the overview of what the whole figure 1 should look like. And so then what you're doing is saving this figure 1 as a PDF or an image that you can then put onto your digital PowerPoint template for your genes and disease project. So where do you find all this information? Most of this information you've already found. So what we did earlier for a pre-assignment for this week was complete that post-activity worksheet for the exercise we did with Pompe disease, where everything we did in class was focused on Pompe disease. And then for the post-activity, you were supposed to find that same information for your particular gene and disease topic. So a lot of the information, like how many transcripts it has, what's the length of the mRNA, all that kind of stuff, you've already found, and that information should be on this worksheet. So you should have most of the information you need to start filling in the details for your figure 1. So for most of the images, like I said, you can just create a shape in PowerPoint, but for the diagram with the transcripts, so remember that was where it said like GAA at the top, and then we had 3 transcripts, so we had 3 green lines that say NM whatever, and then those are exons. So this isn't something that you create, rather this is something that you pull from NCBI. So what I recommend is using some kind of screen capture tool to grab that image from NCBI and then insert it into your figure 1. Make sure you're referencing that appropriately, so include that in your references in the header for your figure 1. I personally use Macs. So for Mac, what you want to do a screen capture is if you press Command plus Shift plus 4, what that'll do is your cursor will turn into this little bullseye, and you can click and drag whatever section you want to capture. So if you click Command Shift 4 and then put that bullseye, drag it to create a box around this image that you want, what it'll do is turn that into a screenshot image. It saves it automatically to your desktop. So you'll have a screenshot there that you can then insert into your figure one. PCs are a little bit different. They usually have a print screen button or something similar. If you're not sure how to do it specifically for your operating device or system, if you Google it, you should be able to find it. But that's how you would obtain that image for that particular section of your figure one. So good luck. Again, you have most of the information already. It's just a matter mostly of putting it into that format that's shown here. And if you have questions, let me know and I will help you how I can.